Welcome back, Indiana Virtual. As promised, here is a video on the virtual lab activity for lesson 0301, cell division. Okay, so we're going to go ahead and dive right in and start reading a little bit of background information. Okay, it says in this virtual lab activity, you will be observing the cell cycle in the tip of an onion root. The root tip is responsible for the downward growth of the root and is one of the regions in the plant where cells are actively dividing and growing. Because of this, the root tip is an excellent system in which to observe the entire cell cycle, including the processes of nuclear division, mitosis, and cell division, cytokinesis. Okay, so before we click on anything further down here, we're going to look at the instructions. Okay, now this says virtual lab instructions, but it's really a little bit more about background information than anything else. We have more instructions later on. Okay, so in terms of some background information, the roots of onion plants are used for observing cell division. One, because they're easy to grow. They're always actively dividing, so it's really easy to see the different stages of mitosis. They're easy to look at underneath the microscope. They can be manipulated and prepared in such a way that makes it super easy to look at them. And they typically have very few chromosomes. Onion species typically have just a few chromosomes, and they're usually really large and easy to see underneath the microscope. Okay, there are 3 regions to the tip of an onion root. Okay, there's the root cap, that's the protective layer. The region of cell division, so here is where that interphase, prophase, metaphase, anaphase, telophase, and cytokinesis are all taking place. So there is your major cell division going on. And then in the region of cell elongation, the cells are getting bigger, so the plant is getting bigger, the roots are growing longer, okay, but no division happens in the region of cell elongation. Okay, that's a really important thing to remember. Maybe a post-lab question somewhere, that cell division only occurs in the root tip, in the region of cell division, nowhere else within the plant. Okay, now says, for this lab activity, you will be examining a section of an onion root tip under a compound light microscope. These slides were prepared by slicing the roots into thin sections, mounting them on glass microscope slides, staining them so the chromosomes are more visible, and then covering the specimen with a cover slip. Okay, and here's some images of what you're going to see in the activity. Here is a cell interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. Okay, so the lab report. The lab report has our typical sections: title, objective, hypothesis, data. We have 2 trials. We have cells in part 1, cells in part 2. We have some observations that we're going to record. We have a data analysis section that has a part 1 and part 2. And then we have a conclusion and a post-lab activity or post-activity question section. Okay. So before we get into this, I am going to go to our classroom or our Google site, and I'm going to open the template over here as a Google Doc. I've added some extra information on our Google Doc template to help you a little bit. So I would suggest that you use this Google form rather than the simple template from Buzz. Okay, so let's begin here. So the title. The title, remember, is your name, my name, and whatever you want to call the activity. The objective here is, we are trying to observe the cell cycle. We're trying to observe the cell cycle in onion root tips, and we're trying to learn, what do the stages of the cell cycle look like? What does interphase look like? What does prophase, metaphase, anaphase, telophase, and cytokinesis look like? So that's our objective. We are trying to help you be proficient with recognizing these images and what is happening in the stages of the cell cycle. Okay, now for hypothesis, we need to go back to the activity and start reading through this introduction so you can figure out, what are you even supposed to write a hypothesis about? Okay, in this laboratory activity, you will be examining a section of an onion root tip under a compound light microscope. These slides were prepared by slicing the roots into thin sections, mounting them on glass microscope slides, staining them so the chromosomes are more visible, and then covering the specimen with a cover slip. You will classify each cell based on what phase it is in, and then count up the cells found in each phase. Use those numbers to predict how much time a dividing cell spends in each phase. So right there, that last sentence is what your hypothesis is about. Okay, you are to predict how much time a dividing cell spends in each phase. So if we go back to our lab report, for your hypothesis, I would like you to make a prediction on the length of those phases. Okay, so maybe, for example, you think interphase is going to be the longest phase of the cell cycle. Or maybe prophase is going to be the longest phase of the cell cycle. Maybe you want to go the opposite spectrum. Maybe you want to say cytokinesis is the shortest phase of the cell cycle. Either way, or maybe you want to rank them. Rank them in which one you think is going to be the longest to which one you think is going to be the shortest. Okay? But that's what your hypothesis is about. You are trying to predict the length of time spent in each phase of the cell cycle. Okay, once you have your title, objective, and hypothesis, we are now ready to collect some data. Okay, so let's go back to our lesson. Okay, we've got a little bit more of intro information here. The life cycle of a cell is ongoing, but can be divided into 5 major phases that can be seen and identified relatively easy. The image below give examples of what you might see in each stages of the cell cycle. So here we are. We are to roll over each phase to learn more about interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. Notice when we roll over each image, we're given a description below. Okay, now here, just a little tip for you. All of these descriptions and images can be used on our lab report. Okay, I'm going to skip below this data just for a second to the observations section. Okay, you could record your observations as to what happens in each of these cells. What can you describe in interphase, prophase, and so on. The images here give you a really nice description of what you are looking for or trying to observe in each of those, which you can use for your observation descriptions on your lab report. Okay, so now we're going to move forward. Okay, so now we are in part 1 of our data collection. So we'll scoot back up here. What we are going to do is in part 1, the virtual lab is going to provide us with an image. We have to classify that image as one of the stages of the cell cycle. And once we're done, we're going to record those numbers here. So if we go back, and I will do a few of these for you, or with you. Okay, this first image, okay, this is a picture of interphase. And hopefully you can remember that from the previous slides. And you can always back out of the activity and maybe take a screenshot or a picture of those images so you can use here. Otherwise, you can also take your best guess, and eventually you get the hang of it and figure out which cells are which stages. Okay, so this is a stage of interphase. So I'm going to click on interphase, and if it pops up right here and says interphase, and it adds my tally over here, then we know I'm right. I made it. So yes, that is a stage in interphase. Okay, now here we have some clear X shapes. We have some clear chromosomes that are visible. The chromosomes are not in the middle. The chromosomes are not pulled apart. They're not separated into 2 sections. So that tells me that this is a prophase stage. Okay, we'll click on to the next one. Okay, now if you remember, this looked just like the very first one. So that's an interphase as well. So we're going to continue to do this and tally these up until we get to the very end of part 1 of 50 cells. Now it takes a little while. We have 50 cells in part 1, and then we have to do it all again and have 76 cells in part 2. Okay, so you might be thinking, well, what if I guess wrong? So let's just take a guess. Well, let's say this is cytokinesis. Oh, Did you see that? It tells you incorrect. Please try again. Well, maybe it's telophase. Nope, not quite. How about anaphase? Hey, there we go. That one is right. So no worries if you get it wrong. You can have as many guesses as you need to. Just the more times you guess, the longer it takes you to get through 50 cells. Okay, so you're going to continue on until you get through all 50 cells. And once you get through those 50, you're going to take these numbers and you are going to record them over here. Okay, once you're done with the first 50, it's going to roll you right into part 2, where there's 76 cells. You're going to do the exact same thing and document those numbers here as well. Okay, now once you're done with that data collection of part 1 and part 2, then record your observations if you haven't done so already. And then we're going to get into the data analysis. So the data analysis here, I created an extra data table for you to try to help this be a little bit easier, because sometimes students can struggle on this part. So hopefully what I've done here for you has made it a little bit easier than what Buzz provided. Okay, so what we're going to do in part 1 is calculate the percentage of the cell cycles spent in each stage using the following calculation. So here's our calculation. We're going to take the number of cells in a given stage, divide it by the total number of cells counted, and then multiply by 100 to get a percent. Okay, and that's the percentage of the time of the whole cell cycle that's spent in that one stage. Okay, so first thing what we have, these two columns are the exact two columns from the table above. You literally are going to copy your data from this table into this table. Okay, then in this column, you're going to add column 1, column 2 to there. Okay, so for example, if we had 11 cells here, 12 cells here, our total number is now 23. Okay, let me come up with, well, actually, yeah. Okay, I'll just get rid of that. Okay, so same thing. However many two numbers you have for prophase, this number plus this number makes that number. Okay, now when you're done and you add up all those numbers, they should equal 126. If they don't equal 126, then something's off. Okay, now this final column is where you have to do a little bit more of the percentage calculation. Okay, so you're going to take the number of cells in the given stage, so that would be this number here. Okay, so all right, let me try to do a little bit of math. We had that. Okay, so how we'll do this calculation here, it's the number of cells in a given stage, so it's going to be 23 divided by the total number of cells counted, so 23 divided by 126. Okay, and don't forget our order of operations. And then we're going to multiply that by 100. Okay, so 23 divided by 126 multiplied by 100 is 18.3. So we're going to round to the nearest whole number. So this is 18%. Okay, we'll do the same thing with prophase. Add up the two, part 1 and part 2 of prophase, get the total number of cells in prophase, and do that same type of calculation. The total number of cells in prophase divided by all of the cells times 100 to get the percentage of the time spent in prophase. Okay, now keep in mind, I just made up this number just to give you an example. Okay, once you've done that, part 2 now. Part 2 is where you're going to use your percentages in part 1 to create a chart that represents the time spent in each stage of the cell cycle. A lot of students like to do a pie graph here. It's easy to take a pie graph and divide it into the different sections of percentages because the entire pie graph completed as a whole is the 100% of time. Okay,